All right, thank you so much for having me. It's great to be back here. I think I was a participant a few years ago, and it's amazing to see the great talks. I enjoyed the session today actually because it really focuses on technology and the role of technology in changing care moving forward. And I'm going to really make that point one more time through my talk about how AI and technologies are enabling better personalized medicine. That's really going to be the focus of the talk today. So the mission of our institute is really to try to rethink medicine to make it much more personalized. Our vision is that medicine can and will be personalized, much more personalized in the future than what it is now. But I think really the crux of my talk and the slides I'm going to present is really focused on the fact that we need to understand the biology of every individual. Everybody is different. Everybody gets cancer, gets Alzheimer's, diabetes in different ways based on their genes, based on their exposures, based on all kinds of different things. We need to understand the disease within each patient much more so than what we've done in the past. What we need is essentially to build a map of people's disease. This is an example of what a map could look like. This is a very complex map, and this map is going to be very, very complex. But as you'll see in the next few slides, we think that we have the technologies now to really start building this map, to assess clinical samples from patients, whether it's cancer samples or blood samples or other kinds of biopsies. We can use technologies to assess these samples and really capture the underlying biology of patients in a very, very detailed and sophisticated way. So these are the technologies I'm going to talk about today, ranging from spatial techniques to assess tissue at single-cell resolution, genomic analysis of DNA and RNA to understand the genes and the mutations that are expressed within individual patients, as well as techniques such as being able to grow cells from patients in a dish and essentially make avatars of people's disease and then test all kinds of different drugs on these avatars before treating the patient. All these technologies, maybe 5-10 years ago, we'd think of them as science fiction. They're really starting to be real and starting to be applicable to patients, and I'm hoping to be able to give you a sense of that. And none of this would be really feasible without AI, which is kind of the glue between all these technologies that allows this integration and analysis across tens of thousands of patients to be able to get insights and then understand how we can predict outcomes in the future. And that's really the different ways to think about our work is to think about pillars. We have these technological pillars, this layer of AI that's really kind of gluing everything together with the goal of improving how we deliver medicine and make medicine as precise as possible. So I'll start with sequencing of DNA and RNA, which has been the workhorse of precision medicine in the past several years. At Y Corner Medicine, we decided to really take a very broad look on the DNA and the RNA. We're using a technology called whole genome sequencing, which is sequencing patients' DNA, whether it's cancer DNA or germline inherited DNA, from beginning to end. We're sequencing from the beginning of a genome to the end of a genome. That's about 3.4 billion nucleotides that we're sequencing for every patient. And the goal is really to try to understand every single mutation. We really want to leave no stone unturned to understand essentially disease and the driver of a disease within each patient. We're doing this across many different types of conditions, cancer still being one of the critical conditions where we applied DNA sequencing, but we actually had some very large programs at Y Corner Medicine trying to apply whole genome sequencing to all kinds of different conditions. For example, heart disease. In heart disease, cardiovascular disease, we actually have sequenced thousands of patients with whole genome sequencing, again, sequencing of every gene, every mutation, what's between the gene, and so on. And we're seeing actually very great potential for this kind of technology. We're seeing ability to better diagnose, for example, inherited arrhythmias, better understand early onset atrial fibrillation, and return results of progenitor risk scores, which is a new way of thinking about genomic information, which is not to say that individual mutations are very important, but it's more of a combination of inheriting lots and lots of mutations that adds up to a high risk of developing certain conditions, like atrial fibrillation. And so this is a new tool in genomics that's actually very powerful, that assesses the entirety of a genome almost, and can report reliable information about risk of developing conditions in the future, but also helps in terms of diagnostics. When you have two different potential diagnostics, maybe the risk can tell you whether one is more likely than the other. We are applying this analysis of genomes to cancer as well, and this is just an example here of one patient where we sequenced the entire genome of a cancer genome, the genome of a cancer DNA, basically the tissue itself. Applied a machine learning technique that allows us to understand the entirety of all mutations, the mutational signature across the entire genome. Thousands of mutations, when you combine them together, give you some clue about some of the underlying phenomenon active in the tumor. In this case here, it was a concept called BRCAness, which some of you, I'm sure, have heard about BRCA mutations. Patients with BRCA mutations will develop cancer at a high rate, but it also means that there are some therapies that are going to work better in those patients because the BRCA mutations is damaging the ability of cells to repair the DNA, and there are drugs that have been designed to be able to essentially push the cancer cells over the edge. When you have such a weakness in the cancer cells, essentially they're very susceptible to certain types of drugs. So it's also a therapeutic opportunity. The issue is that many patients have BRCAness but don't have a BRCA mutation. But we know now that the analysis of the entire genomes can give us information about BRCAness and therefore the likelihood that they'll respond to certain therapy, like platinum, PARP inhibitors, for example. And this, the patient here on this slide is an example of a patient that had a gynecological cancer, did not have a BRCA mutation, but according to a machine learning analysis of the genome, had this BRCAness concept, phenomenon, the signature. And accordingly responded really well to platinum therapy. As you can see here, the tumor melted away as a result of knowing that information about the tumor itself. Not just that, what's really interesting about whole genome sequencing is because we sequence everything in the cancer from beginning to end, we can actually look for mutations that explain the BRCAness phenotype. In this particular case here, we actually found that there is a fusion. There's basically a break between chromosomes and a fusion between two half chromosomes that's creating a new gene in the cancer cells that's connected to BRCA in the sense that one of those genes here is known to interact with the BRCA gene itself. So that's essentially an explanation why you have BRCAness in this one patient. So it's really this full picture that gives us all these clues about the disease and helps treat those patients really, really well. We're going beyond that. We're also trying to think about how we combine new drugs. We've pioneered the use of AI to come up with ability to choose combinations in patients with cancer. The future of cancer therapy is not to treat cancer patients with one drug at a time because resistance occurs really quickly. It's really to combine multiple drugs at the same time and really kind of hit the cancer really hard with no way of escaping the therapeutic pressures. But it's been very hard to come up with combinations because there are many drugs available and how you combine them is just, essentially there's a lot of possibilities. And that's where we think that AI can help, and we have some proof of concept data showing that. We put together a machine learning, a deep learning-based analysis of RNA and DNA in cancer cells. And we taught this model how to pick combinations of drugs that are predicted to work together really better than individual drugs one at a time. And this is what this deep learning network looks like with multiple different layers that combine information about the drugs, about the target, about the patients, all of that to eventually make predictions. We haven't applied this yet to patients, but we're seeing now that the predictions that come out of these algorithms are actually really, really good. We're able to validate the prediction in cell lines or in tumor organoids. I'll show you more about tumor organoids that essentially show that we can pick combinations that are very synergistic when we test them in the lab. So I think that's really, really promising. One more layer that's really important is just go beyond DNA. DNA and DNA mutations, especially in cancer, is really focused on the cancer cells. But we know now, after many years of working on cancer, that a tumor consists of billions of cancer cells, but also billions of non-cancer cells, immune cells, blood vessels, all kinds of additional cells. And there's a lot of data showing now that this non-cancer cell populations are really important when it comes to understanding the biology of every individual cancer. So we developed techniques to be able to analyze cancer tissue at single cell resolution. Well, now we can look at tissue and understand every single cell and be able to tell what cell that is. What kind of cell is it? An immune cell? What kind of immune cells? A macrophage, a T cell, a CD4 T cell, CD8 T cells, and so on. And essentially, you go from really having this kind of measurement here where we're analyzing lots of different proteins in cancer tissue to this map here where every cell, that's actually a real biopsy from a patient. This is a cancer of B cells, B-cell lymphomas. And you see actually the green cells here are the cancer cells, but you see all of the other cells in the biopsy that are not cancer. You have CD4 T cells, CD8 T cells, you have macrophages. You actually really see what a cancer looks like. And as I said, it's not just cancer cells, there's a whole ecosystem in there. So we can use technologies now to assess tissue at this kind of resolution, and it's very powerful. So powerful now that we can essentially take this picture here, analyze, count the different immune cell populations, the tumor cell populations, look at proximity between different cells, look for different patterns, combine all of this information with information about the patients, the age, BMI, and so on, and be able to predict response to therapy with a tremendously high level of success. This is a survival plot here with predicted patients, essentially patients predicted to respond to therapy versus patients predicted not to respond. And as you can see, the predicted responders are doing really well, and the predicted non-responders are doing very poorly. The therapy in those patients with B-cell lymphomas here is chemotherapy. It's actually been very hard to select patients for future response for this kind of therapy. But the machine learning analysis here, looking at different factors of a tumor, just based on the picture that I showed you, as well as information about the patient itself, really is very powerful when it comes to prediction of response. It's just another example of the power of machine learning, but combined with the right measurements and the detailed analysis of every individual tumor. Just another slide here, which is a different technology that we have, which is, as I said, the ability to grow tumor cells in a dish from individual patients and essentially get a lot of tumor chunks that for every patient that essentially kind of recapitulate the disease in every patient. The reason for doing this is that we want to be able to test all kinds of therapies in those tumor avatars, if you want, here before we treat the patients. So just essentially we get more data points per patient, and we're more informed when it comes to treat the patients. So that's something that we have at Cornell, but it's also very unique. It's very unique in the sense that we apply this to every cancer patient. It's pan-cancer by definition, and so we're growing this. These are examples of these tumor chunks growing in a dish for different types of tumors, bladder cancer, stomach tumors, melanomas, and so on. We combine this with high-throughput robotics to be able to test all kinds of different drugs and combinations within each individual patient using this robotic system here. And this here is an example of a screen. This is for a colon cancer patient here where we're testing all these drugs. You can't read them, but it doesn't matter so much. It just gives you a sense. These drugs here at the bottom are very effective at killing the tumor cells of this one patient here. They may not be effective at killing the tumor cells from another patient with colon cancer, but this is really an assessment per patient. So essentially think of it as additional data points here that we can combine with DNA analysis, combine with this single cell measurement of tissue, really to essentially see how, essentially we can pick drugs that are likely to work in every patient. We've done such screens now for so many patients that we're starting to be at a point where we can think about maybe not having to do the screens, because we have a lot of data, lots of data points for many patients. We have the DNA, the RNA. And so we're starting to be at a stage where we can essentially take one new patient and then place that new patient in the context of all of the similar patients with the same DNA, same RNA, with the same disease, and essentially be able to say, well, those patients responded to this therapy, right? So therefore, this new patient here, because it's so similar to those older patients, may actually respond as well. For some therapies, like for example topotecan, we actually can see that those predictions are actually quite reliable. The predictions are actually quite good. They're not good for every drug. We probably need to do more screens, but they're starting to be really good. So it just gives you a sense as well of where the field is going. It's a combination of experiments, and at some point you have enough data and you can build this kind of models that seems to be very good. Just my last slide to really emphasize that AI is really becoming so critical for everything that we do. We have work in our institute focused on using AI for reproductive medicine, for IVF. We do work in thinking about other kinds of biomarkers to collect from patients, such as voice, for example, changes in voice over time that we think are predictive of certain types of conditions or maybe able to help us diagnose some conditions really well. And again, drug response, which we are using organoids or these avatars to essentially get more of, so that we can build machine learning models that are predictive of our drug response in the future. So AI is critical to everything that we do. As I say, it's really the glue that is combining all these measurements here. We're really looking forward to using more AI in the future for everything that we do. I'm going to stop here. Thank you for your attention, and I'd be happy to take any questions.
Future of Care is New York City's premier community for healthcare innovation. To learn more, subscribe to our channel and visit our website at futureofcare.nyc.